Hello, and thank you for joining us today for Hamilton Thorne's webinar, Advantages of Using Lasers in Embryo Biopsy Procedures. The webinar can be viewed in full screen mode, so if you choose this setting, the button is displayed at the top left side of your monitor or in your control panel. There are also a few videos in this presentation, so we recommend that you close out any additional applications on your computer to provide for smoother viewing. The webinar will be conducted in 3 parts, followed by a Q&A session at the end of the webinar. You can provide your questions throughout the webinar, and during the Q&A session, we'll have our panel of experts answer as many questions as time allows. Any questions that we are not able to address during the live session, we'll send a follow-up email to you directly with an answer. The first session of the webinar will be led by Dermot Douglas Hamilton, CTO and Senior Vice President of Research and Development at Hamilton Thorne, who will be providing an overview of our laser systems and embryo biopsy procedures. After Dermot's talk, we'll take a quick poll and share the results with the audience. Next, we'll have our guest speaker, Dr. Behr, IVF Lab Director at Stanford University, who will be giving a talk on embryo biopsy methods and technologies. And with that, I'm going to turn the talk over to Dermot Douglas Hamilton to provide an overview of Hamilton Thorne's laser products and embryo biopsy procedures. So with that, Dermot, I turn it over to you.
Good morning. I'm going to be talking about the use of infrared lasers in embryology, and in particular some of the ones that we have developed at Hamilton Thorne, and talk a little bit about the type of light we're using and what the effects of the laser are, and then setting the background for the practical clinical use. Can I have the next slide, please? The laser is used in single pulse applications for laser-assisted hatching on the zona pellucida of the embryo, and for biopsy in removing part of the zona pellucida so that a blastomere can be removed. That would be typically at the 3-day stage, and also at typically around the 5-day stage for trophectoderm biopsy and removing the trophectoderm cells from the embryo. So we'll discuss those in a later point. The machine that we're using, the particular laser, is called the Lycos. I'll show you roughly how that works and how we use it. We'll then discuss the effects on the embryo and the effects of the particular kind of infrared light we're using. And finally, I'd like to summarize with the effects of the multipulse that we've developed, which is particularly applicable in trophectoderm biopsy. Could I have the next slide, please? The infrared lasers that we have are used for assisted hatching, blastoma biopsy, and trophectoderm biopsy. And we've just shown pictures here of the typical single pulse hole generated in the zona pellucida for assisted hatching. For blastoma biopsy, we usually remove a larger region. This can be done with a flap, leaving a flap so that there isn't so much of a large hole left in the embryo. And finally, in trophectoderm biopsy, where the trophectoderm is herniated out, we use the laser in order to weaken the bonds of the cells holding the biopsy onto the embryo. Could I have the next slide, please? Now, I'd like to show you a brief image of exactly how this works. In this case, we can start it here, and what you're going to see is a small red circle representing the laser, and a yellow or orange ring around it, which represents the distance at which the zona pellucida will be liquefied. You can see it moving here. The laser stays in the same place. The embryo is moved usually on another pipette, and once the area has been carved out, the pipette is put in and the embryo is removed, generally quite easily because it's not very sticky at this stage, which is the 3-day stage, and now that's complete. May I have the next slide, please? The instrument that we're going to describe is the Lycos. This is a system in which a standard objective, 40x, and a numerical aperture of about 0.6, so that gives you a nice bright image and very clear. Contains a laser, and it also contains a beam that tells you what the location of the laser focus is going to be. And in the next slide, I can show you a picture which gives you a schematic of how this system works. The laser is over here and fires a beam down, which is reflected onto a dichroic mirror. That's a mirror that reflects the infrared light but doesn't reflect the visible light. So that goes up to the target, which is at this point, and the visual image of the target returns down here and goes to the camera and eyepiece. At the same time, we have a little source on this side which produces a red beam that goes off the other side of the dichroic and goes down towards the camera and the eyepiece. And by adjusting the position of this red beam, we can identify the exact spot that the laser is going to be focused at. The laser and the visible are focused at the same point. No adjustments are needed, so that makes it a relatively trouble-free piece of arrangement. May I have the next slide, please? Now, the laser light that we use is at 1416 nanometers, or 1.46 microns, if you prefer it that way. We normally use it pulsed between 100 and 1000 microseconds, generally around 200 microseconds. And the power while the beam is on is always 300 milliwatts. The real point about this beam is that it's very strongly absorbed by water. In fact, water appears to be something rather like almost black ink to this particular light. It gets absorbed in the water, and when it's focused, it superheats the water. The result is that the zona pellucida is in fact liquefied by a tiny little jet of extremely hot water. As soon as the beam is turned off, it cools back in a few microseconds to the same temperature as the rest of the system, so that it doesn't affect anything nearby. Now, the way that this light works is that the photon of light is absorbed directly by the water molecule. And the reason I'm emphasizing this is because it's slightly different than what some other lasers do. It's absorbed by the water and makes the water get very hot. The water has an oxygen and hydrogen, and they're connected by sort of strings. You might think of them as elastic onto each other, and they begin to vibrate. When the photon reaches them, they stretch and they bend, and that is how the energy gets into the water. It's then very, very, very quickly changed into ordinary heat, and that's what has the effect that we normally work with. Now, I'm just going to point out that the absorption of the beam is peaked at just about the region where we are. That's 1.45 microns in that region. Therefore, we absorb the energy efficiently, and it has a maximum effect. It also travels a sufficient distance. It goes like about 0.2 to 0.3 millimeters. That's far enough for the kind of effects we're trying to do. So it's a very suitable radiation for what we're trying to do here. Now, the thing I wanted to describe next was what actually happens when this light is absorbed by the water. And so what I've drawn here is a picture of the laser beam coming in over here, focused down. You must imagine this as a cone, down to a region where it's focused, and then, of course, it escapes out on the other side. And I've drawn this with straight lines to make it easier, but in fact, of course, they're not straight, they're curved. When the beam comes in during a single pulse, it heats up the water, and you can see the amount of heat by these contours, 20, 40, 60, and very rapidly going up to above 300 degrees in the center. So you might say, why doesn't the water boil? Well, it doesn't boil because it doesn't have time. It just has a rapid effect on anything that is in this neighborhood. Notice that this green lozenge-shaped area, can I go back one on that, is in fact at about 100 degrees, and it makes a region which is long and rather thin and quite sharply defined. Now, when that is directed at the edge of a embryo, we can see an example of the kind of cut that's produced. Here you can see that it makes a pretty sharply edged cut in a bovine. Actually, this is a bovine oocyte. It's not a bovine embryo, but the zona pellucida is very much the same and very similar to water, to a human, excuse me. Here we have three trenches of the same type cut in the edge of the zona by that same system, and you can see that the width is about the same as that of the lozenge that I showed you before. The hot region cuts the embryo. Now, a second application of this is in working with trophectoderms. And if we can make this work, perhaps somebody would make that on. There. Okay. Now, what's going to happen is that the video is going to show you somebody pulling off the herniated trophectoderm out of the embryo on about a 5-day level. And now this region here is the laser, and the laser is going to move slowly across here, giving single shots. When there's a shot, it twitches. You see that? A slight twitch. Let's do it again. There'll be another one here, and then another one. Each twitch weakens the bonds between the biopsy and the rest of the embryo. And at the end of it, one is able to separate the biopsy with a minimum amount of time and therefore a minimum possible problems due to that. Now, an alternative to that is that one can use a system in which one is multipulsing the laser. Everything I've mentioned to now has been single pulses, but in doing the trophectoderm, it would be an advantage if we were able to use multiple pulses and move the join between the trophectoderm biopsy and the embryo rapidly across the position of the beam. So that's what we have developed. And in this, we can select the power and the number of shots per second that is produced and the length of the pulse of the laser. We can select those provided that the average temperature, the average power, excuse me, is below 10 milliwatts. That's a pretty low power, but that's actually more than we need. In most cases, we can do what we need with just 1 milliwatt, and that's the number which we recommend using. Under the regulatory rules, we have in fact got the clearance for FDA, and we have the CE mark for MDD. So our system is legally able to be used on clinical, and the result is that we have more effective and rapider dissection of the biopsy from the embryo than in the previous single pulse case. I'm going to show you a brief movie here, which is of, I'm sorry, we have a problem. Here we go. Okay, now we're going to show you the repeated pulse, multi-pulse system for a mouse embryo. And you can see that just moving it across like that causes the separation. This is, of course, a very simple and very delicate embryo in which it's relatively easy to do this, but it works just as well using it for human and for other species. The actual power used in this was below 1 milliwatt, and it produces an effective cut that is simply done. Now, just to go back on what the system does, I remind you that it produces intense local heat for a very, very brief period. During that time, the temperature reaches up to around 300 degrees, but it doesn't boil. As soon as the laser is turned off, it cools rapidly down. If you use multi-pulse, it cools down practically to the initial temperature in between each pulse. So the pulses are independent. When we're doing this, we can weaken intercellular bonds, and we can therefore do the separation of the trophectoderm very simply, and it can be done rapidly. And that's really the main advantage that we're trying to provide here. So all of those applications are clinical and they are available. We have a number of other applications in the research field. One of them is the excision of the ICM, and that's used by people in stem cell research. We also have had a number of people interested in nuclear deletion for transfer and for organelle deletion, which we can also do using this system. So to summarize, the applications in the clinical field are for laser-assisted hatching, laser-assisted biopsy, typically at the 3-day, and for trophectoderm biopsy at the 5-day state. And what we find is that the system is safe. It's been approved by the regulatory authorities. It's very specific because you can fire at exactly the region you want, and it's very limited in time. The effect is on only when you're firing it, usually by pressing the foot pedal, and it's effective. And based on the reaction that we have seen in the past few years, using the laser for these purposes is probably the recommended method of choice. Thank you very much.
Thank you, Dermot, very much on your great talk. So at this time, I am going to launch a poll. So here we are. So the question is, do you use lasers in the following procedures? Assisted hatching, blastomere biopsy, day 3, trophectoderm biopsy, all or some of the above, or none of the above? Please choose one. Okay, great. I'm going to close the poll now, and I'm going to share the results. Looks like a lot of you use all or some of the above. That's great. Okay, so at this time, I'm going to turn it over to Dr. Behr of Stanford University, who will be giving a talk on embryo biopsy methods and technologies. Dr. Behr, please proceed with your presentation.
Thank you very much, Lisa, and thank you, Dermot, and hello, everyone. It's my pleasure to talk to you today on embryo biopsy methods and technologies. And I'm going to be focusing this next 15-20 minutes or so particularly on laser and biopsy technologies for pre-implantation genetic diagnosis or pre-implantation genetic screening, more than assisted hatching, which has been around, as most of you are aware, for 20+ years. First being applied with partial zonal dissection and acid tyroids, and more recently, I think, by most people using lasers. If I could have the next slide, please. So we all are aware that genetic material may be obtained from gametes, at least oocytes, day 3 embryos, as well as day 5 or day 6 embryos at the blastocyst stage. And my bias, as you can see here from my first slide, is that I believe there are significant advantages to biopsying embryos at the blastocyst stage over day 3. And I'm not going to talk much about a polar body biopsy, but I think there's enough debate out there as to the value of this technology that I'm going to stick to using day 3 and day 5 biopsy for my comparators. But first and foremost, one of the salient advantages of biopsying the embryo at the blastocyst stage is that the only embryos that are being biopsied are those that are embryo transfer quality. It is very relevant nowadays where array-based technology is being used, where there is essentially a cost per embryo after a certain point that both on a monetary or fiscal level as well as a practical level for the laboratory, biopsying only those embryos that are transfer quality makes sense. With day 3 biopsy, approximately 40 to 60% of the embryos you biopsy do not make it to the transfer stage, and hence money is spent and time is spent on biopsying embryos that don't quite make it to transfer stage. As well, over day 3, trophectoderm biopsy offers the opportunity to get more robust results because we can get more cells removed safely from the embryo than can be done on day 3. So we need to consider that not only is there a time savings, there's a potential to get more robust results with a trophectoderm biopsy. Additionally, it is thought that the trophectoderm cells pose more of a barrier with regard to mosaicism, or less of an issue with regard to mosaicism, than what is witnessed in day 3 embryos when there may be a high degree of mosaicism present in the embryo within blastomeres. Additionally, depending on the location of one's laboratory relative to the molecular genetics laboratory, the requirement of cryopreservation or vitrification may indeed be necessary with day 5 biopsy, and some may consider this a disadvantage, others may consider this an advantage. However, if results are not able to be obtained within 12 to 24 hours of the biopsy, then vitrification or preservation of the blastocyst would be necessary if the biopsy is done on day 5. If I could have the next slide, please. So one of the questions and the technical aspects about doing trophectoderm biopsy is how can we create a herniation, or should we create an opportunity for the embryo to prematurely hatch such that there is a herniation for us to grab onto, as you've seen in the videos previously shown, for the trophectoderm biopsy. And it is my strong bias and strong opinion that one should do either a trough, as Dermot showed before, or a complete hatch, as you see here on the bottom left panel, on day 3 between blastomeres, such that you can encourage hatching on, herniation on day 5, on the morning of day 5. There is, some debate about whether a complete hatch is necessary, i.e. penetrating all the way through the zona, as I show here, versus a trough, as we saw previously. And I would say that you should base that decision on how robust your blastocyst growth is in your laboratory. If you're getting very, very early blasts traditionally on day 5, I would suggest making a complete hatch. If your blasts are typically fully expanded on the morning of day 5, or at least a significant number of them are, then I would just coax the embryo out with a small trough that will essentially make a weakening area on the zona, encouraging the hatching at that particular site. Next slide, please. So one of the issues that one has to consider when doing this hatching on day 3 is, where is the embryo going to hatch? Of course, on day relative to the structures in the blastocyst, that is, we know where it's going to hatch because we just made a hole where we want it to hatch. One of the downsides and one of the considerations needs to be is, what do you do if the inner cell mass begins to hatch at the area that you made your trough or your breach? And remember, on day 3, we don't know where the inner cell mass is going to form, so it is somewhat of a random formation, and I would argue that if what happens in the picture I'm showing you here, i.e. the breach is at the area of the inner cell mass, you're unlucky. But from personal experience, this happens the minority of the time, just randomly speaking. Statistically speaking, this is not the most common area for the embryo to hatch with or without a breach. So what I do in the case of this is either make another hole 180 degrees from where the inner cell mass is, so that we can go in and grab some trophectoderm cells and remove them from the embryo that way, or potentially remove the embryo completely from the zona pellucida and biopsy the embryo that way. I will warn those of you who have good blast growth in your laboratories that occasionally an aggressive embryo will escape the zona completely, and that does not pose a significant hurdle for the biopsy. One just has to be very careful about anchoring the embryo with the holding pipette, holding onto a significant piece of trophectoderm cells with a biopsy pipette, applying the laser at the cell junctions where the embryo, if you will, is hanging off the biopsy pipette, and then simply rub your biopsy pipette onto your holding pipette, which will essentially sever the trophectoderm cells in the area where you applied the laser off the rest of the blastocyst. So it's much like a PZD approach, for those of you who are aware of what that is, where you make a slit in the zona by essentially placing a needle through the zona and then rubbing it on your holding pipette. The similar type of approach can be applied to a fully hatched blastocyst. Another technical aspect to be aware of, and again Dermot alluded to this in his talk about weakening junctions of cells. It's important to recognize when you apply the laser on the cell body of a trophectoderm cell, that cell essentially will be destroyed by maybe boiling water inside the cell. If nothing else, it may denature the DNA in that cell. So it's important to recognize when you are removing trophectoderm cells, the number of intact cells that you are removing. We may take a big chunk and stretch trophectoderm cells, but if we're firing all along the body of those cells, the cells that come out that have been fired on are not really going to be accessible to the probes for amplification, and hence you may have less cells than you think. So that's an important feature to pay attention to, which relates to the last two bullets on the slides, which are to really emphasize looking at where the cell junctions are and trying to apply the laser between the cells versus on the cells themselves. Occasionally, this cannot be avoided. Remember, we're dealing with a three-dimensional structure, and there may be cells beneath the cells we're firing on that may indeed get some laser on the body, but that is not a problem given the number of cells we have to work with. Next slide, please. So I want to talk about the philosophy of the timing of biopsy, and maybe more the philosophy of, why we go to blastocyst. And when I started trophectoderm biopsy, and actually when I started blastocyst transfer many years ago, there was, a lot of emphasis on making sure we identify the viable blast, the blast that has a identifiable inner cell mass and a multicellular trophectoderm layer. And there are many patients who make several embryos of that quality, realizing that still the implantation rate of blastocysts on day 5 or day 6 is still around 50%. So it's clear that although we may be reducing or shrinking the denominator, our power to select the baby maker with morphology alone is not sufficient to pick the single embryo that's going to make a baby. So the philosophy that I'm referring to is a bit of a mindset difference, which is to think of going to blastocyst as an opportunity to eliminate embryos that have arrested on day 3. So to avoid waiting for the perfect blastocyst, which may form in the morning of day 5, in the afternoon of day 5, and again in the morning of day 6, doing 3 biopsies and 3 freezes, our approach now, and seems to be working exceptionally well in my lab and several other laboratories now, is biopsy anything that on the morning of day 5 that is not arrested. As you can see here in my second bullet, we do not wait for the expanded blastocyst anymore. Anything that's a morula or beyond will get biopsied. Clearly, a morula, you cannot remove multiple cells. You need to decompact the morula, as I'll talk about in my next slide, and remove a single cell. So clearly it is more towards the cleavage stage biopsy. But when you look at the greater good for the patient and the practice, doing this seems to be the right way to go, primarily because it gives you information, genetic information, on your whole cohort of embryos. Otherwise, if you wait for the fully expanded blastocyst, particularly if you're doing a fresh transfer on day 6, which I advocate in my programs because of excellent results, it does not give you the opportunity to get the genotype on all your embryos. Clearly, if you're doing a preservation cycle where all the embryos are going to be cryopreserved for transfer on a subsequent cycle, you can biopsy as many times as you want and still be able to get the full information on the cohort of embryos. And I do realize and understand that this opportunity for fresh day 6 transfer is really only available to those programs who are within a stone's throw or an hour or 2 flight away from a PGD or PGS molecular lab. If I can have the next slide, please. So what about the biopsy media? It was clear and apparent when we do day 3 biopsy, cleavage stage embryo biopsy, that the use of calcium magnesium free embryos, particularly in those embryos that have begun some compaction, is advantageous. A compacted embryo makes it very difficult to remove a blastomere, increasing the risk of lysis or tearing of the membrane of the blastomere. However, this does not seem to be the case with trophectoderm biopsy. I don't believe that calcium-magnesium-free medium actually poses a harm to embryos. I have not seen any data to suggest that. And from my personal experience, when I first moved to trophectoderm biopsy, I was using calcium-magnesium-free medium with excellent results. However, it does not seem to be necessary, and there does not seem to be any advantage, at least from the sort of seat of the pants feel. Therefore, we use modified HDF now, or ambient air buffered medium, to do trophectoderm biopsy. As I mentioned previously, when you are doing morula, which essentially is a very highly compacted, multicellular cleavage stage embryo, calcium-magnesium-free media makes a huge difference, and makes for a much more trouble-free biopsy, even though you are removing a single cell. It provides for an opportunity, as I mentioned previously, to get the genotype on all the embryos available for transfer potentially on the morning of day 5. I think for any vendors out there or any media makers out there, I would make a call, shout out for ambient air buffered blastocyst media. It just makes sense to me that if you could biopsy an embryo in a medium that is osmotically and oncotically more similar to the media that the embryo has been sitting in for the last couple of days, it may be beneficial, although I have no direct evidence or data to prove that. If I could have the next slide, please. So we are moving, as I mentioned before, to the era of molecular genetic results and no longer relying on fish. And I thought I would just put one slide in here just to address, for those of you who still may be doing fish, even with trophectoderm biopsies, which is how I started. Fixing trophectoderm cells or blastomeres is not a standard embryology skill. Certainly wasn't something that I grew up with in the field, and it's a skill that I had to acquire. Several problems with fixing, and this may be beating a dead horse because most of us have moved on from this. However, it does provide an opportunity for certain types of genotypes, if you will, for sex-linked diseases and things of that nature. The two problems I'm referring to is that if you don't fix well, the results can be significantly compromised, which is not a good feeling to think that something we weren't trained in is going to affect the results. And secondly, you need a second space to do the fixing because of the odors that the fixative generates. This typically is not done in the IVF lab. On the other hand, for doing tubing, for the molecular approaches, the array-based technologies, pipetting we've all been trained in and something we're comfortable with. If you get the cell in the tube, you typically get a result. It's not contingent upon how well you pipetted. Although clearly if you put too much media in a tube, that can potentially affect the results. And last but not least, this is something that we can do in our IVF lab right in the hood that most of us have, and don't have to go anywhere, just maybe have to get dressed up a little bit more than usual. And you can see on the bottom right panel, this is a piece of trophectoderm that has been removed after biopsy, showing little sort of straggly cells at the bottom where cell bodies may have been lysed. However, you can see at least 2 or 3 intact cells at the top of that biopsy piece. Okay, next slide, please. So what about managing the sticky trophectoderm cells, which can happen, particularly if the embryo is completely hatched and you have to rub, as I described earlier, the biopsy piece or the sort of dangling embryo on the holding pipette to erode it off the trophectoderm. There are 2 approaches that can be used. One is something that you can do tomorrow or today in your lab, and that is load a PVP drop in your biopsy dish and simply wash or aspirate, rinse, if you will, your tools in the PVP prior to doing the biopsy, which should essentially lubricate it to a point that will reduce the stickiness. Additionally, and I've done this in a research mode, not for clinical applications, is have your tools be silicone coated. I'm not familiar with where this can be done for clinical applications, but certainly having some coating on your pipettes can alleviate that stickiness problem if it becomes a problem or is a problem for one to do. It is not something I do routinely, but in anticipation of a sticky hatched embryo, we may include this technique prior to embarking on the biopsy. If I could have the next slide, please. So this is just a nice picture showing how one would approximate the embryo with a biopsy pipette, aspirating. Here you can see just one trophectoderm cell, and how I would apply the biopsy or the laser in this embryo would be, you can see very clearly here where the cell junctions are, to pick a cell junction maybe close to where the zona pellucida is, and down at about 9 o'clock, and try remove those 3 to 5 cells on the outside of the trophectoderm. Excuse me, on the outside of the zona pellucida. Next slide, please. So in summary, I would like to mention the topic that Dermot mentioned about the multipulse system that is ideal for herniating trophectoderm during trophectoderm biopsy. This really has, I would argue, revolutionized the process for us in the laboratory. It enables a very smooth, rapid, safe approach to removing the biopsy piece off the trophectoderm. Typically, one would aspirate the biopsy piece into the biopsy pipette, and then enable the laser with your mouse, and then apply the foot switch for a shot, and then reactivate the laser and repeat this step multiple times until you've dislodged or weakened enough bonds to actually dislodge the trophectoderm. Biopsy piece from the trophectoderm. What this new system allows is to enable the laser, as you see there on the left side of the image. You can adjust all the power and have full flexibility on the pulse duration and the milliseconds and power, etc. And once you have enabled the laser, you don't have to take your eye off the eyepiece. You can see where the laser is going to fire. You have both hands on the joystick, and you simply swipe, if you will, both hands across the laser beam, and within a fraction of a second or a couple of milliseconds, you have essentially dislodged or weakened the trophectoderm bonds enough such that you have a smooth biopsy and a nice piece of trophectoderm in your biopsy pipette, as was seen in the video. So I would encourage you to try this. I love mine. And I think for those of you who do trophectoderm biopsy, you'll find this a significant sort of shot in the arm, if you will, in terms of making the process a lot easier and safer for the embryo. I believe that is it. If I could have the last slide. I'd be happy to, I believe, Lisa, you're going to do another poll before we do the Q&A session.
I'm going to jump right to the Q&A session, actually, but okay. That was great. Thank you so much, Dr. Behr. And at this time, we are going to begin our Q&A session. I'll read your questions to our panelists, and they'll respond to as many as we can. As mentioned, any questions that we do not answer, we'll provide follow-up correspondence. So we have been getting several questions, so I thank you with that. And I'm going to open the mic to both Dermot and Dr. Behr, and I'm going to start off with this question. I believe this is for Dr. Behr. What size biopsy pipette for blast biopsy, which is expanded and not hatched?
I use a smaller internal diameter biopsy pipette for trophectoderm than I do for blastomere. The standard blastomere biopsy pipette is between 35 and 45 microns internal diameter. I use about a 25 micron internal diameter biopsy pipette for trophectoderm cells because, as you saw in the picture, they fold nicely, or in the videos, they fold nicely and can go in to the biopsy pipette quite easily.
Great, thank you. This could be for Dermot as well. How often should we calibrate the scale to measure the size of the hatch site?
Well, the scale can be calibrated just with a reticle which is on the microscope, and it won't change, but I would recommend for good practice that you should calibrate it once a week. You'll usually find it hasn't changed at all.
Okay, great.
I can tell you our calibration has never changed in 7 years or more than that, but that's
Exactly.
a good point, Dermot. It doesn't change.
Yeah.
Great. So another question, is there an ideal droplet size in which the embryo can be placed during assisted hatching biopsy procedures to minimize the high temperature related risks?
I can answer this, but I think Dermot can as well.
Sure.
I think the heat and cooling is so rapid that I don't believe that the drop size would be like the ocean relative to the size of an embryo, as small as you can make it. So we typically hatch in or biopsy in 20 to 25 microliter drops. But I don't know, Dermot, you can chime in here. I don't believe
you're exactly correct, actually, Barry, because it's the region that you're heating is so tiny compared to the droplet size that it's like being in the ocean. So that the heat will rapidly get dissipated in the whole thing, and you won't see any change.
So I don't believe there's any change in the drop temperature regardless of the size of the drop, as long as it's big enough to fit the embryo.
That's correct. A very, very slight increase, which is negligible.
Right.
Great. What percentage protein do you recommend for micromanipulation?
I use the standard protein concentration for embryo culture across the board for micromanipulation, same as for culture.
Great.
Which in my case is 10% volume to volume using a serum substitute.
All right. What is the laser power for a blast biopsy, Dermot?
It's we typically use a power on the laser pulse of 300 milliwatts, but the pulse, if we're using multi-pulse operation, is generally 1 milliwatt. That is to say, we have a few pulses spaced out, so the average comes out to 1 milliwatt. And that's the kind of power that's used. It has the effects which I've shown you, essentially independently from each pulse.
Great, that's terrific. There are some questions here that are about specific our lasers, so I think maybe we could take some of these offline. Dermot, what is the best power value to use here?
I strongly recommend using the power value at 100%. The reason is that the more intense the beam is, the less long you have to leave it on, and the smaller the amount of heat that will diffuse anywhere other than exactly where you want it to be. So for best safety, it's best to use it at 100%. But for very weak embryos, for example, if you're dealing with mice or something like that, you may want to reduce that number and still find that you get exactly the cutting you want. That's the answer I would give.
Sure. Dr. Behr, here's an interesting question. How would you troubleshoot when a blastocyst collapses during the biopsy?
Yes, that's a very good question, and it is quite common that when the blastocele is breached by the first fire of the laser, that the blastocele may indeed collapse. And this is important relative to the trough or the hatch hole that is made on day 3. It is for that reason that I prefer a trough or make a very, very small hole so that you can create some tension. When you grab onto your trophectoderm piece and you have a big hole, if you pull hard on that trophectoderm piece, you will be able to pull the whole blastocyst out of the hole. This problem is exacerbated when the blastocyst collapses. So my recommendation is make the smallest hole possible on day 3 or trough. And if the blastocyst begins to collapse and you have a small hole, you still will be able to have sufficient tension to be able to separate the cells without pulling the whole blastocyst out. It has happened where the embryo will, we will hatch the embryo completely by tugging too hard, and that is not a problem for us. Completely hatched blastocysts work fine. They are slightly more difficult to manage. However, it is not a clinical an issue really with respect to outcomes. So again, small hole. Keep an eye on the blastocele as it collapses so you can create some tension. You can always hold on to a piece of the blastocyst with a holding pipette if you like.
Great. What type of micropipette do you use for transferring TE cells to lysis buffer tube?
I use standard stripper tips, flexipets, whatever the device du jour is that you use in your lab. You should use a fresh one per piece to ensure that there's no carryover, but just a standard, embryo, as I mentioned in my talk, standard embryo manipulation pipette. You can use a smaller bore to ensure that you have the minimal carryover with respect to volume into the tube.
Great. And for non-PGD day 3 ETS, would you hatch day embryos in their culture drops, or would you move them to new media? Are there any risks for keeping the embryos in the same media they were hatched in?
I'm not aware of any risks or any changes that the laser would create in the media that would pose a risk to the embryo. In fact, I think one of the major advantages of the laser system is it's so rapid and easy that it does not require you to make embryos go from the hot tub into the swimming pool, if you will, changing media and changing the environment for the embryo, so it has to adapt to something differently. Although brief, you can hatch the embryo in your culture dish and put the embryo back in the incubator within less than a minute, and not change the environment substantially at all. So again, advocate leaving the embryo alone as much as possible.
Okay, great. And perhaps you can both answer this, but definitely Dr. Behr. What is the proper hole size for blastomere biopsy?
Well, there's, my opinion is to create a flap. Dermot mentioned this, although the video that we saw showed an opening that is the size of the blastomere. And what will happen for sure in those embryos is on day 5, you'll either have a completely hatched or a very large herniation coming out of the embryo with a large hole. So my approach is to make one, medium power slit, if you will, in the zona, such that I can create a flap, much like a PZD hole, and access the blastomere that way without having to make a hole the diameter of my biopsy pipette.
And I would recommend one thing there, which is that in making any holes of this sort, use a lot of small pulses rather than one or two really big ones. Because if you make a big pulse, the heat will convey itself into the other blastomeres. If you make a lot of small pulses, it won't get anywhere other than where you're trying to hit.
Great. Dr. Behr, can you discuss your day 5 versus day 6 pregnancy results? Any problem related to performing biopsy on day 5 and ET on day 6?
Yeah. As I mentioned before, my personal preference and bias, and actually better outcomes, drove me to do day 5 with day 6 transfer. We have not, I've published on this, 15 years ago on the results on day 5 versus day 6 pregnancy rates, and it is well established now that a day 6 embryo after biopsy does very well in the uterus, in a fresh uterus on a superovulated woman right after IVF. So I mean, I think spewing numbers is not that helpful, because there are a variety of variables that contribute to success rates, but I can tell you that my best success rate with PGD, or PGS I should say, PGD is not that easy. We don't have as many options, if you will, to do this fresh on day 5, day 6, but it certainly can be done. With PGS, the best results I've had per cycle initiated, which is really what patients care about, is doing a biopsy on day 5 and a transfer on day 6 and freezing anything that's normal on day 6.
Great. Dermot, you could start with this one. What is the idea behind fixing the laser point?
Fixing the laser point. The laser has to be focused at exactly the same point as the objective. In other words, you must have the same focal position. When you focus the microscope, your laser must be in focus when it's visibly in focus. So in that sense, we fix it that way. If that's the meaning of the question, that's what we do. In terms of the position of the laser inside the field of view, we do this by fixing it because we like to be as close to the optic axis as we can in order to get the maximum best image available. And it's also convenient if you're trying to move the embryo across the beam, for example, to have a fixed point of reference.
Great. What portion of day 5 blastocysts do not herniate after day 3 breaching of zona, and what do you do in that circumstance?
You know, again, this is very lab specific, and it really depends on, you know, how many blasts one's lab sees expanded on the morning of day 5. But it is inevitable that regardless how robust your lab is, there will be some embryos that are slightly delayed metabolically or kinetically, resulting in a sort of, you know, a non-homogeneous pool of embryos, let's call it that. So even if you have good blast development and you have most of your embryos herniating, you're going to have one morula or one early blast. And what we do in that case is literally either enlarge the hole if there's nothing, you know, if the trough did not actually breach the zona. If we made a trough, we make a hole and actually grab onto some of the trophectoderm cells about 180 degrees away from the, well, the opposite pole of the embryo from the inner cell mass, and grab onto them and tug some out. In those cases, you almost inevitably will get blastocele collapse. So it's important that you don't, you know, bore a huge hole in the zona, so that you still can create some tension and make it easier to essentially dislodge the trophectoderm cells off the embryo.
Okay, and which is better, biopsy of early blast on day 5 and fresh ET on day 6, or frozen ET?
Boy, that's, you know, that's, it really depends on how good your freezing is. I would argue that if you look nationwide, look at pregnancy rates nationwide, as much as people talk about how fresh is as good as frozen, when you look at all the labs, the 400 and something or 500, close to 500 in this country, and then the thousands across the world, freezing does not help embryos. It adds a potential opportunity for damage. It's an additional variable. So my argument would be, if you can avoid freezing, you should. I know not everyone agrees with me, and I respect other opinions, but in my hands, if we knew that freezing was standard, like every time you froze you were going to get the exact same result, then I would argue that maybe freezing would not be as much of a problem. In addition, patients, no matter how good your freezing is, patients' material have different cryo tolerances. So there may be 2 people with the same quality embryos. One survives 100% and one survives 60%. So again, there are too many unknowns, too many variables to argue that freezing should be the standard for everyone. I would say in specific circumstances, if you have a fantastic cryo program and you can identify those patients that will freeze well, then, you know, go ahead, be my guest. But I would say, you know, to preach this to everyone is a mistake, because results demonstrate, international statistics show that freezing does compromise embryos overall. Maybe not in every specific program, but overall, freezing is not benign.
All right, terrific. Well, we do have lots more questions, but we can't get to them all. So at this point, I'm going to conclude the Q&A session. And actually, that is going to end our webinar today. And I want to thank all of our speakers for sharing their time and insights with us. So thank you, Dr. Behr, and thank you, Dermot. We especially like to thank the attendees who joined us today. If you missed any portion of the webinar, or you feel a colleague might benefit from the talk, we will be sending out a recording of the webinar for you to listen to at your convenience. If you have any follow-up questions, and I know we didn't get to them all, especially some related to our laser specifically, please feel free to direct them to Dermot Douglas Hamilton so we could discuss our lasers in more detail. And any general webinar questions, please feel free to contact me directly, Lisa Rivero, and there's my contact information. And thank you again for your time. and we hope that you could join us for our next educational webinar. Thank you very much.
Thank you.
Thank you.